Hello everyone, let's get started. Today we're going to be talking about cellular structure and function. In order to understand the structure of various types of cells and the function that those cells perform, we have to first understand how cells are visualized, and that involves microscopy. There are a couple of names that you should be familiar with regarding the history of microscopy. The scientist named Robert Hooke is credited with being the first person to look at tissue underneath a microscope and describe what we now call a cell. Hooke lived in the 17th century, and he fashioned a very primitive microscope for himself. A microscope that was capable of magnifying things by about 30 times. Hooke examined a piece of tree bark underneath his microscope, bark from a cork tree, and in that bark he saw what he described as hundreds of individual compartments, and of course we would now refer to those compartments as cells. It was Hooke who gave them the name cell because when he saw them under the microscope, they reminded him of little rooms, like the rooms that he had seen inside of monasteries where monks live. Hooke had seen the little sparsely furnished, very plain, individual rooms that monks would reside in monasteries, and that's exactly what he was reminded of when he saw each individual little compartment in the cork bark. So he called these compartments cells, and we still refer to them as cells today. Now, another scientist whose name was Antony van Leeuwenhoek, a scientist who lived in the same time period as Hooke, was able to take Hooke's microscope and advance it a little bit further. Leeuwenhoek was able to increase the magnification of these primitive microscopes about 10 times. And Leeuwenhoek was interested in looking at types of tissue other than tree bark, other than plants. So he set out to examine a lot of animal cells, including human cells, blood cells, sperm cells, and so on. He also examined different kinds of algae and even some bacteria that he was able to locate in the environment. So Leeuwenhoek expanded on Robert Hooke's original work and helped us to understand that there are actually a variety of cells in nature, and they're all different in terms of their structure and their function. Now, in the 21st century, microscopy has advanced quite a bit since the days of Hooke and van Leeuwenhoek. The most commonly employed type of microscopy is light microscopy, and you have probably handled a light microscope at some point in your career as a student. Light microscopes use visual light in order to view specimens, and they are much more powerful than those primitive microscopes would have been. Light microscopes can magnify anywhere from about 50 times up to about 2,000 times, depending on the microscope. Most light microscopes magnify to a maximum of about 1,000x, and that would be under what we call the oil immersion lens. Remember that when you want to figure out how powerful a microscope is, you take the power of the lens and you multiply that by the amount of magnification that the eyepiece provides. The eyepiece is typically 10 times magnification, and the most powerful lens, again on most microscopes, is 100x. That's the oil immersion lens. So that gives a total of 1000x magnification. Much more powerful than what those men were looking through back in the 17th century. Using a light microscope, you can visualize all types of cells. Now you'll notice I have a little asterisk next to the sentence on the slide, and that's just a reminder to us that we cannot see viral particles under a light microscope. They are simply too small. But viral particles are not cells. They don't meet our definition for a cell. Remember, in order to be a cell, you have to be surrounded by a membrane. You have to contain cytoplasm. You have to have DNA. And you have to have ribosomes. And viral particles just don't meet that definition. Now, when you're using a light microscope to visualize cells, there are actually 3 different types of viewing options in the light microscope. There's what we call bright field, there's dark field, and there's phase contrast. Each type of light microscopy gives you a different view of the objects that you're examining. On this slide, we can see 3 different images of epithelial cells. These epithelial cells happen to have come from someone's cheek. In the upper left-hand corner, the cells are being visualized using bright field microscopy. You can see there are 2 cells evident here. The cells are stained a blue color, as is the background. You can see a dark colored sort of circular structure in the center of the cell. That's the nucleus. These happen to be, again, animal cells. If you didn't know that they had come from the lining of the cheek, you could guess that they were animal cells by their shape. They don't have that typical square or rectangular appearance that we would expect from a plant cell. The key feature of this image that tells us that it was taken using bright field microscopy is that the objects, the cells, actually appear darker than the background. So that's how things are visualized using bright field. The objects that are being visualized are going to appear darker than the bright background. Now, dark field microscopy is essentially the opposite. Dark field produces a darker background and lighter cells or other objects. So on this image, you can see 1, 2, 3, 4, 5, 6, 7, maybe 8 individual epithelial cells. Again, you can see the nucleus of each one of these cells in the center, but it has a very different appearance overall when you look between the bright field and the dark field image. Finally, down at the bottom on the left-hand side, this image was taken using phase contrast microscopy. So I think you can see that you get almost a sense of the 3-dimensionality of these epithelial cells when you look at this image. You can see that this cell, for example, and this cell are overlapping each other right here. You just get more of a sense of the 3-dimensional features of these cells when you use phase contrast, and that's what phase contrast is designed for. We often use phase contrast when we're examining living cells, because it allows us to see some of the structural 3-dimensional detail without having to kill the cell and stain it. So bright field, dark field, and phase contrast are all different modes within light microscopy. Now there are two other modern types of microscopy that you should be familiar with. One is called fluorescent microscopy, and the other is electron microscopy. A fluorescent microscope is different from a light microscope because it uses a phenomenon called epifluorescence in order to visualize the specimen. So instead of using just plain old visible light, a fluorescent microscope uses this fluorescence characteristic to excite molecules that are in the specimen. And what happens is those wavelengths that hit the specimen will cause certain parts of the specimen to fluoresce, basically to light up underneath the microscope, and you get a very bright, very unique looking image. Fluorescent microscopes produce images that primarily have a dark background, and the object can be colored different colors based upon the stains that are applied to the specimen. There are lots and lots of different fluorescent stains available now, and each type of stain will bind to different macromolecules in the cell. For example, you can stain a specimen with a protein stain, and it will stick to things in the cell that are made out of protein. You could tag or label the cytoskeleton in a cell using a different fluorescent dye. So there's lots and lots of fluorescent dyes available now, and we can get some really amazing images of very particular cell structures using a fluorescent microscope. Now, fluorescence microscopy, as you might guess, is more expensive to do than light microscopy. The microscope itself is much more expensive, in the tens of thousands of dollars range, and the dyes that you need to purchase are also quite expensive relative to ordinary stains that we would buy for a light microscope. Now, the last kind of microscopy that you should be familiar with is electron microscopy. Electron microscopy is an amazing technique that allows us to see cells and cell structures at an amazing level of magnification. In fact, up to about 10 million times magnified. And the reason it's capable of such amazing magnification is because it doesn't use light to view the specimen. It uses a stream of electrons. Now, there are two very different types of electron microscopes. One is called transmission, and the other is called scanning. Transmission electron microscopy, or TEM, is the version or the type of electron microscopy that allows us to see the internal structure of a cell. In comparison, scanning electron microscopy, or SEM, allows us to visualize the three-dimensional surface of the cell. So you get a very different image depending on whether you're using TEM or SEM. And through the semester, what we will do as we examine cells in different tissues in the body is we will look at images that are taken with light microscopes and electron microscopes as well. On this slide, for example, there are two images taken of skeletal muscle. The image on the top left was taken with a light microscope. The image on the bottom right was taken using a transmission electron microscope. Now, if you look at the image at the top, what you're seeing here are these elongated structures This is skeletal muscle. It's stained this bright pink color because a stain was applied to the cells when the tissue specimen was put onto the microscope slide. It's difficult to see individual cells on this slide. These are not all individual cells, but you can see individual nuclei. Those have stained purple. Muscle cells are different from many other types of animal cells because they don't have that sort of rounded spherical shape that we associate with animal cells. Instead, they are quite elongated and linear. In fact, we don't really call them cells. We typically refer to them as fibers. They still have all the same components that any cell would have. They're still wrapped in membrane. They still have cytoplasm in them. They still have DNA, and they still have ribosomes. And of course, there are additional organelles in them as well. But they're also loaded with different types of filaments that enable the cell to do its job, which is contraction. Muscle cells gather together in order to form these larger structures that make up the muscles that you think of as being your skeletal muscles. They all work together to achieve contraction, and they all lie all pointing in the same direction so that when they contract, the entire length of the muscle will actually shorten. So you can't see a lot of detail in this image other than, as we said, the individual purple stained nuclei in individual muscle cells here. But you do get a sense of the general shape of this group of muscle cells all lying together in a linear conformation. Now take a look at the image down on the bottom. This is the image that was taken with a transmission electron microscope, and this looks very different. You are not looking at a big group of muscle cells here. You're looking inside a single muscle cell, and you're looking at a very tiny fraction of what's inside. I said a minute ago that skeletal muscle cells, or fibers, are loaded with these filaments that are going to enable that cell to contract. That's what you're looking at in this image. You're looking at some of these filaments, again, all stacked up on top of each other. I'm moving the cursor back and forth to indicate how these fibers, these filaments, are aligned with each other, creating this very unusual sort of striped structure inside the muscle cell that we call a sarcomere. So you can see that from this image to this image, the increase in magnification is just tremendous. Whereas we are looking at dozens of individual cells in this image, we're looking at the inside of one cell, and in fact only a small portion of the inside of one cell in this image. We're going to be talking about sarcomeres and about the process of muscle contraction in great detail later on in the semester. On this slide, I have another comparison between a light microscope image and an electron microscope image. But this time, the EM image is taken with a scanning electron microscope. If you start with the smaller image up on the left, this is the structure that I want you to see, this circular looking structure right here. It's stained pink. It's broken apart a little bit over here. That's just an artifact of when this slide was made. This slide is actually a cross section or a transverse section through the oviduct, or what we used to refer to as the fallopian tube in the female reproductive system. This is the tube that carries egg and sperm and brings them together in the female so that fertilization can take place. Now this white sort of spike-looking structure in the middle, this is actually the cavity. This is actually the central tunnel through the center of the oviduct. Remember, this is a transverse section, like a slice of bread through that tube. So you can see there's not a lot of opening inside that tube. The bulk of the tube is made of tissue. And although it's hard for you to see in this image, there are actually layers of different tissue here. You can just make out that they've stained slightly different colors. This layer of pink right here is the darkest. Then there's another layer from here to about here that stains a little more lightly. This again is this very outer layer. This is just some supportive tissue that would have been captured with the oviduct when it was taken for sectioning. The other thing I want you to notice is that around the white cavity there's a dark staining, sort of a line around that white cavity. It's stained a purple color. That's a layer of tissue as well. It's a very narrow layer, of course, but that's the innermost layer. And it's the cells that line the oviduct, and they too are a type of epithelium. So you can barely make it out. You can barely see a little bit of purple color lining that white cavity. That's the epithelium. Then this darker layer here, that's a layer of smooth muscle. And then we have a layer of fibrous tissue on the outside. So you can't tell any details about these epithelial cells. You just can't see them. You can see where they lie. They cover the surface of that white cavity running through the oviduct. But you certainly can't get any information about the cells from this light microscope image. But now look at this image. This is the epithelial lining of the human oviduct, taken with a scanning electron microscope. And again, the scanning EM gives you a surface view of that tissue. Look at the detail that you can see in each one of these individual epithelial cells. So it's as though we're standing in the cavity inside the oviduct. It's as though we're standing inside that white space and we're looking at the wall. Each one of these circular structures is an individual cell. You can see that on some of these cells there are these long hair-like structures coming off. Those are cilia. The oviduct is full of cilia. It's the cilia that move and transport the egg and the sperm down the length of the tube. Amazing, amazing amount of detail with an image taken with an electron microscope. So again, we will be looking at images all semester long, and most of them will be taken with either a light microscope or an electron microscope, although we'll try to throw in some fluorescent microscope images as well. But regardless of how we visualize different cells, we can organize different types of cells into various groups based on what we call taxonomy or levels of organization. And you probably learned these levels at some point in your career. Things like kingdom and phylum and class and order, all the way down to genus and species, we can organize living things into groups based upon where they fit in this scheme of taxonomy. Now, if you look at the largest grouping of living organisms in the taxonomical scheme, the very largest group is called the domain. And there are actually 3 domains of living things on our planet. There are what are called the bacteria, the archaea, and the eukarya. On this slide, up at the top, you're looking at an image. It looks a little bit like a tree. And it's designed to look like a tree. It's what we often refer to as the tree of life. The technical term we use is phylogenetic tree. It's a tree that we use to show not only the different types of organisms that live on our planet, but how they are related to each other evolutionarily. So at the bottom of the tree of life, at the very base of the tree, we imagine that this is the first life form that lived on planet Earth. It's what we often refer to as our common ancestor, or sometimes you'll hear it referred to as the last universal common ancestor, or LUCA. This is the first cellular life form, whatever that was, at the base of the tree. In other words, it gave rise to all of the other life forms on the planet. The trunk of the tree and the branches of the tree remind us that long periods of time pass through evolution as different types of organisms emerge. So if we follow up the trunk of this tree in this drawing, you can see that there's a branching that occurs right here. This happened very far back in the Earth's history. A group of some of those original organisms took their own path evolutionarily and became what we now refer to as the bacteria. Sometime after that happened, there was another branching event. Some of the organisms went on to become the archaea, and other organisms took a different branch, a different pathway, and became what we now recognize as the eukarya. So it doesn't matter what kind of an organism you are. If you live on our planet, you fall into one of these 3 groups: the bacteria, the archaea, or the eukarya. And of course, human beings are in the eukarya group. The bacteria evolved first, the archaea evolved next, and the eukarya evolved next, most recently. Now, eukaryotes are in the eukarya domain. And you and I, of course, are eukaryotes. We are eukaryotic. The bacteria and the archaea are both types of prokaryote. And if you remember from some of your previous courses, prokaryotes are single-celled organisms, and they have no organelles inside of them, so no nucleus, no mitochondria, none of the organelles. They're very simple organisms, single-celled organisms, and some of the oldest organisms on the planet. Now, unlike the prokaryotes, eukaryotes do have organelles. Our cells do have a nucleus and mitochondria and endoplasmic reticulum and so on and so on. Remember though, it doesn't matter if it's a prokaryote or a eukaryote. There are some things that all cells share: plasma membrane, cytoplasm, DNA as the genetic material, and ribosomes to make proteins. Now, just a reminder, Organelles, if you translate that word, it means little organs. So just like you have a heart and lungs and kidneys, you have large organs inside your body. You have little organs inside your cells. You have nuclei and mitochondria and lysosomes and peroxisomes. Those are organelles, and they are, by definition, surrounded with membrane. All organelles have at least one membrane around them, and some have two. Ribosomes are not organelles, therefore, because they aren't surrounded by membrane. It's important to know, though, that some source materials, some textbooks, for example, will refer to ribosomes as non-membrane bound organelles. Just so you know, that strange definition is out there. We're not going to use that definition, though. For us, in this class, if it's an organelle, it has a membrane around it. So ribosomes are not organelles. Now, at the bottom of this slide, we're shown a drawing, a labeled drawing of two types of cells. There's an animal cell here on the left and a plant cell here on the right. These are eukaryotic cells. They are loaded with organelles. You can see in this drawing that the animal cell is given that sort of generic round spherical shape, and the plant cell has much more geometry to it. It has squared off edges. This one kind of looks like maybe a hexagon. But the idea is that plant cells have rigid, linear edges, and that's because plant cells are surrounded by a cell wall. Plant cells have cellulose around them. It's what makes their cells much sturdier than animal cells are. Animal cells are softer and squishier because they have no cell wall. I also wanted to say just a couple of things about the fluid inside of cells. You'll notice that I have two terms listed on the slide. I have the term cytosol and the term cytoplasm. You should know the difference between these two words. Cytosol refers to the fluid inside of a cell. Cytoplasm refers to the fluid and the things that are dissolved in that fluid. So in cytoplasm, there is water, there's cytosol, but there's also everything that's dissolved in that water. Lots of nutrients, protein, carbohydrate, lipid, and of course the organelles are sort of floating in the cytoplasm. So remember, cytoplasm, it's not water. It's a gel-like material. It's much thicker and more viscous than water is. The other thing to remember is that the drawings down at the bottom of this slide are meant to represent very generic animal cells and plant cells. In reality, when you look at a multi-celled organism, like an animal or a plant, the individual types of cells in each of the different tissues in the body are going to have very different shapes, very different sizes. Eukaryotic organisms are extremely complex, and different types of cells exist so that they can perform different functions for the organism. Animal cells, generally speaking, are smaller than plant cells, but both types of cells vary tremendously in their shape. And of course, that shape is directly connected to what function that cell is going to perform.